I had sent this to Steve Day to load onto Canvas, and he's at a meeting, and apparently, I don't know what it was that was going on, but I had a lot of trouble with the email system and the whole intranet system last night, and evidently he did too, so it didn't get loaded up onto eBay. And I thought that I had sent it this morning when I saw his email, but maybe I got involved in trying to figure out how to get you all 4 extra credit points, and it distracted me from what I thought I was going to be doing. Okay, has everybody turned in anything they're going to turn in so far? And I think we're passing around a piece of paper that you can put your name on for attendance. Today, we're going to talk about the mitogen activated protein kinase system, or the MAP kinase pathway, which generally responds to protein receptors for growth factors. And the prototype there would be the insulin receptor, but clearly other proteins as well, such as EGF, FGF, nerve growth factor, BDNF, brain-derived growth factor, glial growth factor, all of these cells that make growth factor proteins that now can activate their receptors. So these are all going to work by phosphorylating tyrosines. And I just showed you what those 2 binding sites are that will recognize those tyrosine phosphorylated proteins. So what were they? What domains on what protein? What domains? SH2 domains, and the other one was called, it's obvious. What is it binding to? Tyrosine phosphates, right? So it's a tyrosine phosphate binding domain. Simple name. So, so what we're going to look at today is our MAP kinase cascade. It has a series of proteins that interact with each other, and they're kinases. So it's a phosphorylation and a phosphorylation and a phosphorylation, 3 of them. And so it's got a sequence of events that take place. We're going to describe some of the mechanisms, their ligands, how they work once they get into, to get them into the nucleus so that they can change transcription. Because that's essentially what this pathway does, is either phosphorylates proteins in the cytosol or membranes that are going to change their function, or gets into the nucleus and works as transcription factors to change gene expression. And we're going to take a look at some non-tyrosine kinase receptors that are going through cytokine receptors that use tyrosine phosphorylation as a mechanism, but are not getting phosphorylated on their own receptor. And if we have time, we'll do some signaling through the NF-kappa B system, and that's a factor for gene transcription, and how that's regulated. So we've got several different regulatory pathways. So the first we'll go through in great detail is this mitogen activated protein kinase scaffolding cascade, and it was first really discovered in and organized well in yeast. So I'm going to show you what we learned from yeast and then took back into looking at mammalian systems. So this is yeast, and yeast are very easy for biochemists to do genetics on. Not so easy to do biochemistry because they come with this nasty yeast skeletal, extracellular skeletal. It's hard to break yeast open and necessarily pull out things like a biochemist might like to do with mammalian cells, but it is easy to do genetics. And the genetics are all based on what you end up with your yeast's phenotype looking like. So for a lot of the regulation of a protein, the yeast people and the Drosophila people have named it based on what they thought they saw when they looked at the phenotype. So in this case, the phenotype is sterility. So you know, yeast can either bud or they can actually mate. And if they mate, there are two kinds of yeast. There are yeasts named A and there are yeasts named alpha. And the A release an A factor that interacts with the alpha protein on another yeast. And then you've got your mating interaction, and you've got two genomes that now can be made into what the future baby yeast is going to look like. So if anything happens along a pathway that is required for this mating process, then the yeast is sterile. So they found lots of sterile yeasts and started naming them in chronological order as to what kind of protein they were missing or was mutated. So you can see that when we begin to look at this, we've got lots of proteins named sterile with a number. This is sterile 20, sterile 11, sterile 7, and a big scaffold called sterile 5. So that's what they had to work with, and then they had to figure out what all of this meant. And so working with these different kinds of yeast, you could combine two yeasts and overcome a sterility defect. And that's how you could determine what each one of these was and how they may interact with each other. Well, this whole pathway starts out with, what is this right here? Something we're all very familiar with now. A G protein coupled receptor and a G protein. So this is their main function of these G proteins in yeast. And this one is the one, the mating factor is either alpha or A, and the receptor is either A or alpha. So that you've got your two sexes. Your sex is either A or alpha. Those are your choices. So, so you've got the G protein interaction. And as soon as this beta gamma is released, it forms a scaffold. And it forms a scaffold because actually it's got a plextrin homology domain so that it can interact with a protein. And the one it's interacting with is this, this scaffold protein called sterol 5. Now, sterol 5 then binds to other proteins, and you can see they're named over here, and each one is a kinase. And you can see these little arrows, each one is phosphorylating the next one along the line. So this is a sequential mechanism by which we are phosphorylating a kinase that can then phosphorylate the next one. So if we start down here at the bottom, here's the one that's our mitogen activated protein kinase way down here at the bottom. This one is called Fuse3. Didn't have to do with the mating factor itself, but had to do with fusion of protein, or fusion of the cell when it after mitosis. So this is a serine threonine kinase, and it gets phosphorylated by MAP-ERK kinase, which is another threonine kinase, but it also can phosphorylate on a neighboring tyrosine. So it's called a dual specificity kinase. Now, most of your kinases are either going to phosphorylate on serines or threonines, or they're going to phosphorylate on the aromatic amino acid tyrosine. And this is maybe the only dual specificity kinase that we routinely find in cells, is this one that phosphorylates the MAP kinase. So you can see we've got two little phosphorylations on here. One is on a threonine. The other one is two amino acids over where there's a tyrosine that gets phosphorylated as well. So that gets phosphorylated by MAP-ERK kinase. Now this one gets phosphorylated on a serine or a threonine by MEK kinase. So this is MEK, this is the kinase, MEK kinase. And this gets originally phosphorylated by the sterol 20. So, so meanwhile, we're doing this scaffolding based on our beta gammas, not our alpha. So this is a beta-gamma scheme over here. And ultimately, once our MAP kinase gets phosphorylated at the serine and the threonine and the tyrosine, or the threonine and the tyrosine, it is going to activate other transcription factors that can go into the nucleus. So this is what we knew from yeast. So once we knew that, then biochemists could go look for homologous proteins that did the same kind of function in mammalian cells. And sure enough, we found all of these. Now let me tell you about the nomenclature. The people that were studying this initially were looking for one or two things. So there was a University of Virginia group that was very interested in how insulin worked. And so they were looking at this from the point of view of a regulation that they called extracellular regulatory kinase, or signaling kinase. And they called theirs ERK. Then there was another group that was at University of Texas, and they were studying it, and they were interested in mitogens beyond the insulin receptor, but these various growth factors, and they named it mitogen activated protein kinase. And so for about 10 years, you could have someone come to give a seminar and they'd start out and their title was yada 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 MAP kinase, and then they'd keep talking about ERK. Or sometimes in the same written paper, they would start out saying mitogen activated protein kinase, and then they'd call it ERK, and then they'd call it MAP kinase. It was very, very confusing. So what we finally have now for our nomenclature is the MAP kinase is a family of proteins, and ERK is one of them. And ERK phosphorylation is by two different proteins, one at 40,000 molecular weight and one at 42,000 molecular weight. And so these two are both called ERK1 and ERK2. And they are part of the MAP kinase bigger family. So I think that way now we've kept the two labs that invented or that discovered these. They both have notoriety. They both have their names in print of what they named them, but we don't have that same confusion as to which is which anymore. So our characteristics of these, the one that's the dual specificity here is the MAP-ERK kinase, which gave credence to both names since they didn't want to make anybody feel bad. They named it MAP-ERK kinase. May seem funny to you, but you know, you don't want to leave anybody out. Now they also, in the yeast field, discovered that there were lots of this very same kind of pathway that would serve different functions. And so, remember over here, the one at the end was named fuse. Well, the MAP kinase at the end for different functions can be different names. So the pheromone that was being released, either A or alpha, set up this pathway that was for mating. And so there, the MAP kinase is called fuse. But if you're in an environment where you don't have any bacteria to eat, you could be starving, and you could have to go through this pathway called KSS. If you were in an environment where you had high osmolarity, salty water, high residue, you have to adjust that with osmolyte synthesis. So O is here. So this is high osmolarity group. And so this is HOG, and that's for adjusting transporters that will adjust the amount of sodium that can get across your membrane so that your yeast don't swell and explode. And here's one for cell wall remodeling, and here's one for sporulation. So you can see the end result of all of these is a different kind of protein that does different kind of signaling. Well, we've got the same kind of thing in our mammalian system as well. We don't have quite as many choices here, but it's all organized by that same scaffolding and goes through the same sequence of 3 sites. So let's see how it starts off in our mammalian cells. So in our mammalian cells, we can start off with a growth factor receptor, and here we've got a dimer. So this is very typical of an insulin receptor. The insulin receptor is the only one of the growth factors that doesn't have to have the ligand bound to it in order to dimerize. It's got connections here with its disulfide bonding, so that you actually have sulfhydryls that are bonded with sulfur-sulfur linkages that hold it together. So here's a phosphate. Once you've got these two arms together, this one catalytically phosphates the other side of the dimer, this one phosphates this side of the dimer, and you can start out with a phosphotyrosine. And then you start building. So from here, you've got a protein called GRAB that has an SH2 group on it that is going to recognize this phosphotyrosine and bind here. Now at the other end of GRAB are two little SH3 domains. And again, this is Sark homology. So it's this homologous to the Sark kinase that is found as a cytosolic tyrosine kinase. And here an SH3 group recognizes parts of the protein that have high prolines, parts of the protein that has prolines. So if you have a group of prolines nestled together, maybe not consecutively, but within a small region, then you are going to call in things that will bind to this, this region. So in the SOS group, you have a group that's got proline, proline, and this is going to be called in by binding to these SH3 domains. So all this little GRAB protein does is connect. It doesn't have any catalytic activity. It just connects tyrosine phosphate pairing groups with something that's going to bind based on proline interactions. So we've got the GRAB binding to SOS. Well, what does SOS stand for? It stands for son of Sevenless, which was a fancy name describing something that occurred in a Drosophila. So the yeast guys and the Drosophila names are these folks are very keen on making fancy, cute little names. Sterile is nothing compared to some of the names that Drosophila and yeast people have concocted based on what they think their cells look like. Here we've got son of Sevenless. That means that it was a mutant that came after something that had less than 7. So I'll show you what that is. It's the eye. So son of Sevenless is from the Drosophila eye, where each one of these units is an ommatidium, and it has 7 proteins in order to communicate. All of these are the axons going out to the brain. And each one of these is responding to light. So here's the number 7 protein over here. It was a mutation that was found that led to loss of response to light in the visual system. So they called this one Sevenless. And there's our ootidium. And it was part of a series. Here's the 7 protein. And this was a receptor for a cytokine that was on the number 8 of that series that was involved in the ootidium. And when they named the mutant after this, they named it BOS. So BOS interacted with 7 and could induce this R7 neuron that was part of the series of regulation so that the ommatidium could respond to light. So they called this one son of Sevenless. They still obviously didn't know what the protein function was, but They simply called it that because in this whole series of events, this was working this way. Now, what does this mean to us? In our mammalian cells, we all have cytokines as well. And we have cytokines that interact with receptors, and these receptors can set up a similar kind of pathway. So, so we do have some homology here, but that's just, I'm just telling you this so that you get the idea of what the origin of the SOS protein was, so that you can help remember it a little bit. So in this case, if we don't have this precursor being induced, we have a malfunction in our rods and cones. Now, what is this? It's the protein that we in mammalian systems call the RAS protein. It was the very first of the small G proteins that was discovered, and it was discovered because it was an oncogene. When this is mutated, it's a very abundant mutation found in cancer cells. So that is to say, if you look at lots of cancer cells and lots of different kinds of cancer and ask the question, what in the genome is mutated, very frequently it's this little tiny G protein, the RAS protein. And it's because our function, one of our main functions for mitogen-activated proteins is to respond to mitogens. What is a mitogen? What does the stem sound like? Mitosis. So they induce mitosis. So mitogen-activated protein kinase. It's activated in response to these growth factor receptors or other things that are called mitogens. Things that will induce mitosis, things that will cause proliferation of your cell population are called mitogens. Mitogen-activated protein kinase. So when you have a dysfunction that now lets mitogen activation run away, uncontrolled, what do you have? Cancer. Yes. So here is our protein that has the function that is going to be right there, right after that cytokine or that tyrosine kinase receptor that responds to growth factors or mitogens. And this is what initiates that whole pathway in our mammalian cells, is our little RAS protein. Well, our RAS protein I've depicted here for you. It has a GDP binding site because it is a small G protein. You don't see what was, if you go back and look at your pictures in your textbook of what your G proteins, that's your alpha protein and your heterotrimers are, it's got a GTP binding region, and then it's got what they call the helical domain, which is another region. And it ends up being about 40,000 molecular weight. Small G proteins don't have that half. All they have is the GTP exchange region. So we call them small because they're only 20,000 molecular weight when you run them on an SDS page. So the small G protein has a spot here for the GDP, and it's got some regulatory switches, just like the ones that I showed you that were going to be activated by the receptors. So here we've got the GDP. The GDP is going to drop off. It's going to be empty, and then a GTP is going to bind. And it's all based on this green switch 1 here, where this little loop is opening up so that we can get the GDP out. So you know the rate limiting step on G proteins is to get the GDP out. That's what the receptor interaction with the heterotrimeric G proteins is doing. It's not causing GTP to bind. It's getting the GDP out so that the GTP can diffuse in on its own. So then that little switch closes again. So now we've got an activated GTP-bound RAS. And that's going to be activated just like we think of as our activation. Now, we get it activated by SOS. So we just had that son of 7 list. that had been now brought to where our cytokine or our tyrosine kinase receptor was. And it was connected there by the GRAB protein. And the GRAB protein now is connecting with SOS, and SOS has interpolated a little part of that protein in here to hold this switch open. So that's how it just inserts right in here, opens this switch, the GDP comes off, the GTP comes back on again, and we've activated our protein. So what we have is GRAB binding to SOS, SOS interacting with RAS in order to get the GDP off so the GTP can bind. So now we've got an activated RAS. And the RAS is binding to the membrane by an isoprenyl group, which is a lipid that's just going to hang on there like a little tail and keep it right there in the membrane. At that point, it's going to initiate that same pathway that we saw in yeast. It starts out with our activated RAS, and then we bind to a protein that is called RAF. And RAF was just sitting in the cytosol, kind of waiting to happen. But it had chaperone proteins around it that would keep it from being activated. So this one chaperone that's binding to this little RAF while it's in the cytosol is called 14-3-3, and it is binding to the phosphosites that are on the RAF protein and holding it together with a regulatory domain. So when the RAS protein is activated with a GTP, this now comes to the surface, and the regulation here is that this gets dephosphorylated, and now this part drops off. So you no longer have these binding proteins now interacting, holding it as an inactive RAS or RAF in the cytosol. So now you've got your little activation loop over here, and this is where the catalytic site is. This is now going to act as a kinase so that it can move along to the next step and phosphorylate the next protein along, which is the MAP-ERK kinase. So now this gets phosphorylated by the RAF. So the RAF now is a MEK kinase, a MAP-ERK kinase. So it's named RAF, but its function is to be phosphorylating this MEK. So this gets phosphorylated. That makes this active. Once it gets phosphorylated, now this activates the MAP kinase at the tyrosine and the threonine. So this doesn't look like it's got a long chain scaffolding like that sterol 5 was for the yeast, but you can see that it does involve making these complexes, and these complexes started out with, in this case, what would be a growth factor or a mitogen receptor on the plasma membrane. So is insulin a mitogen? Sometimes. You may think of insulin as having a main function of going to certain kind of cells that are going to need to take up sugars for storage, like the liver or the skeletal muscle, or take up sugars that are going to be involved in adipocyte regulation. But in other cells in the body, insulin is a mitogen. So yeah, insulin, our prototype receptor that we're going to think about here, is serving as a mitogen. And so it can go through all of this pathway. Now, our runaway part is that sometimes this RAS protein doesn't hydrolyze its GTP very fast. And if its function of GTPase is dysfunctional, then this GTP is going to stay on forever and not be removed. And that's why it can become an oncogene. and be dysfunctional is that it won't break down the GTP to GDP and turn itself off. So it's continuing to start this whole pathway, even though it might not have been originally stimulated by an insulin receptor or some other growth factor receptor. So it's dysfunctional because it can't turn itself off. And that's what makes it an oncogene. Okay, so just for those of you who like to see things operating, this is our activation loop. This is where we get it phosphorylated. It's right here on this protein, the MAP kinase, so that when it gets phosphorylated, right now it's not. Here we've got the phosphates on, and it allows it to be a little switch. So now it can be activated. It's activated only when it has both of those phosphates on. So how does this regulate transcription? If it's going to be a mitogen, it has to get into the nucleus, right? Otherwise, how's it going to do its function? So here's our MAP kinase. It dimerizes. Here's our little phosphates on the tyrosines and the threonines. And at this point now, it can begin to phosphorylate substrates. And one of them that it's going to phosphorylate is going to be called the regulator of signaling kinase, or an RSK. So again, now we're going to another kinase phosphorylation. If you want to know how cells run, you can pretty much guess it's all going to be phosphorylation and dephosphorylation reactions. I mean, that's generally been everything we have talked about. All these little second messengers have to work through some kind of a kinase in the cell. All the substrates are specific for differentiated functions of all those individual cells. So here we've got our mitogens activating something that can pass through the pores. Now, when you've got the dimer formed in the MAP kinase, then it allows a nuclear pore passage as well. So it's hidden some parts that are not going to allow it to get through the nuclear pores. And it will now expose its nuclear localization signals as a dimer. So as a dimer, it can get into the nucleus as well. So we've got two kinds of kinases now that can get into the nucleus. We've got our RISC protein. How big is our RISC protein? If you were to run it on an SDS page, where would you look for it? What's that number there? 90. How big is it? 90,000 molecular weight. Remember, if you don't know what something does, but you can identify it on an SDS page, most people name it based on what size it is, right? All those ACAPs, the ACAP 79 and the ACAP 150, what size were they on SDS page? 79,000 and 50,000, 150,000. So here we've got this 90,000 molecular weight regulator of signaling kinase, RISC kinase. So this one gets into the nucleus, our MAP kinase dimer. It phosphorylates a substrate called transcription control factor, or TCF. And it phosphorylates here. This one, the RISC kinase, gets in. It phosphorylates something called serum response factor, and this gets phosphorylated. Now these are both going to be the proteins that interact with the response element, and it's the response element that gets interactions with the polymerase. Polymerase 2 is what initiates and starts transcription. So the reason this is called serum response element was when you're growing cells in culture, you need serum in order for the cells to grow. But originally, folks in cell culture didn't know what it was in the serum that was necessary. They just lumped it all together and said, we need growth factors from the serum, without knowing what it was. Now we know that you can have insulin. You can have insulin-like growth factor, which is just exactly what it says. It looks a lot like insulin, but it happens to be the protein that is induced by growth hormone in order to promote growth in tissues of the body. It could be brain-derived growth factor. It could be nerve growth factor. It could be fibroblast growth factor. These all end up being in serum, one way or another. So when you put these mysterious growth factors on, that's the same way that we're growing cells even today, is cells need to have growth factors, and we give them serum to use in our cell culture dishes. Either that, or we have to know exactly what growth factors are going to respond to. So this was originally called serum response element because the folks who were studying insulin didn't really know what it was that was causing their cells to grow, but it came from serum. So the serum response element would respond to a protein called serum response factor, which was phosphorylated. Ultimately, it had to be after this pathway of MAP kinase, but nevertheless, it was discovered to be one of the transcription factors that was necessary to get the polymerase to start moving. The other one was this transcription control factor that's phosphorylated directly by the MAP kinase. So you get both of them together after they're phosphorylated, and now they can activate that response element. Yes?
So it requires two of the SRF to activate the SRF?
Yep.
So does the P90RISC just phosphorylate two of them, or do you need two of those?
You can get the two of them to dimerize later on after they're phosphorylated.
Okay.
In fact, I don't think they'll dimerize down here until they are phosphorylated. But they certainly are. They're not going to work. If they're somewhere in the nucleus and they don't get phosphorylated, then they're not going to interact with this response element. So you know the response elements are fairly small. There may be a sequence of only 5 base pairs, but they are coming as 2 at a time. And so almost all of your transcription factors are dimers, because and sometimes they're palindromes. So it would be 1, 2, 3, 4, 5, 5, 4, 3, 2, 1 kind of a situation. Sometimes they're linear, 1, 2, 3, 4, 5, 1, 2, 3, 4, 5. So all of them. The estrogen receptor requires two estrogen receptors bound to estrogen to activate the two sides of the response element. So everything that we generally think of as a transcription is probably two-sided, one way or the other. So then you've got your transcription of your coding sequence over here. So this one just happens to be the gene for Phos. And you know that Jun and Phos now become transcription factors for putting together a whole plan of different kinds of transcription. So you can see that by going through this phosphorylation, it may take a minute or two, but ultimately you're going to end up with transcription of a whole new plan of genes being expressed and making new proteins. So this is a little bit complex, but it's a very important set of pathways. Now, you can get this whole pathway started off not by the growth factor receptor that we just talked about, but rather you can also do this through G protein coupled receptors in 2 ways. So one way, remember how the yeast were doing this? Way back at the beginning, slide number 2. How did the yeast get this started? How did they start their scaffold? From this G protein coupled receptor interacting with the G protein. The heterotrimer dissociates. Was it the alpha that started it?
Beta.
No, it was the beta gamma. So the beta gamma held into the membrane, could start it in mammalian cells as well. Or the arrestin. So after the G proteins have dissociated and left the G protein coupled receptor, and the GERCs phosphorylate these little phosphothreonines and serine patches, then you can call in arrestin. And arrestin can get this whole pathway started with a cytosolic tyrosine kinase called SRC. And again, SRC was discovered as being an oncogene. So this small kinase right here is going to activate this system by now phosphorylating tyrosines. So you can get this activated through G protein coupled receptors or 7-transmembrane receptors, either through the beta gamma or through the beta arrestin. And again, if you go on this website, this AAA website, or AAAS website, the American Association for Advancement of Science, they're the ones that publish Science magazine. They've got all manner of these signaling pathways that you can kind of trace through. Here's the one that goes through a receptor tyrosine kinase with a grab. Here's another little protein called SHIC, and this binds to the sauce. Sauce interacts and activates the RAS by removing the GDP so the GTP can bind. Then we've got our RAF, and there are a couple of different ones. There's BRAF and CRAF. And now you've got your, this is acting as a MAP kinase, and this is phosphorylating MAP-ERK kinase, and that phosphorylates ERK1 or ERK2. But these are not the only MAP kinases. There's a MAP kinase called P38, and there's, and that's, how big is that on an SDS page? 38,000 molecular weight. That can be phosphorylated, and this can take us through some pathways too. Now, interestingly enough, this was going to end up with a pathway that goes down and can phosphorylate Krebs. Where did you hear about Krebs before? What does that stand for? Well, it does start with something that begins with a C. No, it's not calcium. Well, the R and the E stand for response element, and the B stands for binding. So it's something response element binding protein here. I think I heard somebody say cyclic.
I should have said it.
Cyclic AMP. Okay.
Although you're saying
Cyclic AMP response element binding protein. So here we've got some real confusion going on, because when we studied cyclic AMP response element binding protein, we thought of it as responding to a pKa phosphorylation, right? Here it's got a pathway that can go through, in this case it's interleukin 1 and P38, which is the MAP kinase, and that can actually phosphorylate our Krebs. So, you know, we can really get confused if we want to. This now opens up the idea that Krebs not only responds to cyclic AMP, but it can respond to MAP kinase. Now, did anybody start your homework that I gave you last week? Okay. Well, so you're all going to go home and look at that. So look at that and find out what those authors thought Krebs was being phosphorylated by. Okay, now I got 5 minutes left. We're going to switch over here to, this is our growth factor response. This is our tyrosine kinase. This is the pathway that we just talked about over here. This is our G protein coupled pathway. Here we've got some interactions with beta gamma that can initiate this whole thing, and we can get this whole pathway started that way. But cytokine receptors have a brief modification of this, because instead of having the tyrosine bound to the growth factor receptor itself, it's binding to a kinase that then interacts with the cytokine receptor. So here's our tyrosine kinase receptor and their growth factor. So this is the outside surface. This is the one little transmembrane helix that goes through. This is a tiny, itty-bitty little site on the inside of the membrane compared to this great big extracellular surface. So extracellularly, it's binding, in this case, to EGF, epidermal growth factor. And when EGF, which is a big size protein, binds, it closes off these two sites and allows the interaction on the outside of the membrane. And if you want to look at the chemistry here, this is the hormone, essentially, but you can see the hormone is as big as a protein. So you don't want to just get in your little head that all these small molecules, these drugs and hormones are small. Actually, FSH and LH, luteinizing hormone and follicle stimulating hormone, are big dimers of big proteins. So they're 60,000 molecular weight. Here you've got something that's big too. And so when that happens, you get the two of these dimerizing, but the dimer part is on the outside. And here you can see FGF receptors, and the FGF itself, once it binds here, it really takes up quite a bit of space. It's more like a protein-protein interaction, but it allows this dimerization to take place. So our cartoon here is showing us dimerization, and then we have, in our tyrosine kinase receptors, we have the catalytic subunit as part of the intracellular domain. And it phosphorylates, once it gets dimerized, this side can phosphorylate on a tyrosine over here, and this one can phosphorylate on a tyrosine over here, and we can start out with phosphotyrosines. And once these get activated by phosphorylating their little activation lip or their activation loop, now they can phosphorylate other tyrosines along their very same chain. And depending on which ones get phosphorylated first, and there is a certain order for many of these, then you can get other things that can come in and bind to these phosphotyrosines using either phosphotyrosine binding protein domains or SH2 domains. So here we've got what can now be a scaffold, because now after you've phosphorylated some of these, you can get proteins like Schick or GRAB to bind to these phosphotyrosines. So this can really get you started on a pathway where you can begin building, and you see that there's some other proteins that could conceivably bind here as well. So they can either bind sequentially or they could bind together. So this really gets us going on that pathway that runs through the MAP kinase from this dimerization step. So here we're back to what does our insulin receptor do? Our insulin receptor calls in a protein called insulin receptor substrate that can bind through one of these PTB domains and interact with other proteins, because once it gets phosphorylated, it's near this active catalytic subunit. It can phosphorylate sites on this insulin receptor substrate. This is number one. That implies to you that there's other ones. And then they can call in other signaling proteins. The one that we talked about started out with a GRAB or a Schick, and it started using an SH2 domain that could now call in that protein that series that we just saw. But this insulin receptor substrate can start a whole other kind of scaffolding situation, because now it can interact with a kinase called PI3 kinase that can begin to interact with other kinds of kinases that we'll talk about later at another time. Or it can interact with those proteins that are going to move those glucose transporters into the plasma membrane, so you can let more glucose into adipocytes or skeletal muscle. So you can see how lots of things can happen just from having that one phosphorylation. And here's the insulin pathway, and you can look at that. So I think what I'll do is save the cytokines and the NF-kappa B for not next time, because Tom Lau's going to talk to us about another signaling pathway next time, but the following time when we talk about multiple kinases that will begin here. So don't feel upset that we might have missed something today. We'll begin with this again later.